Dr. Yue-Wern Huang does research at Missouri S&T around aerosol health related to electronic cigarettes, drug delivery that targets genes in cancer treatment, and tissue regeneration focused on wound healing. He's going to present specifically today about aerosol health. Now, since he was a teenager, Yue-Wern has been a landscape photographer. I asked him what is, he just said, I'm a landscape photographer. Well, I wanted more than that. I said, tell me what your favorite landscapes are to photograph. And this was his exact response. Spectacular mountain ranges, tranquilizing lakes surrounded by wildflowers, meandering streams with glittering reflections of fall colors, and peaceful morning glory on prairie lands. Now, I don't know about you, but I can visualize each one of those with the way he said that. Thank you for nodding your heads. So, Yue-Wern, I'm not sure which I want more, to see these photographs or to hear more of your prose. So, thank you for that, and let's welcome Yue-Wern Huang. Oh, there's one.
Thank you for the introduction, and thank you for the invitation today. I'm not going to talk about my photography, but in 10 minutes, I'm going to go through 3 stories of my research project. And I have a lot of opportunities to work with S&T people and other institutes for other research topics. But today, I'm going to focus to the core of my lab's core research. Okay, the first thing is related to the pandemic. At the beginning phase of the pandemic, much is unknown about how pathogens are being transmitted. Okay, and we know that the contact transmission is one of the ways, but how about airborne? So we basically create a model, and in 2 conditions. One is we assume patient with moderate viral load. The other is the patient with a heavy viral load. And after they cough, what happened to the virus? Okay, so at the point of emission, for people with moderate viral load, about 10% of the droplet contain virus, and for the heavy load, it's close to 70%. How about 10 seconds later, the bioaerosol, the droplet size change shifted to the smaller range, and about 60% of the virus containing droplets remain airborne. How about 10 seconds later, in terms of the vertical distribution, we found significant amount of the virus floating within the half a meter of the vertical range. How about person with heavy virus load speaking 1 minute with or without mask? So the take-home message here through this model is mask wearing can reduce 95% of the viral copies as well as the virus containing droplets. And in terms of vertical distribution, we still see a lot of viruses floating within the half a meter of the vertical range. So my suggestion to you is next time when you flight, try to avoid the airplane aisle seats, because that's where people stand up and then exhale and then talk and then flip their iPhones. And I think the poster need to be far apart a little bit next time. Okay, how about 6 feet rule? Okay, so we assume the exhale behave like a turbulent round jet, and then do the modeling. And we found 10 seconds after the virus was exhaled, it can travel about 2.64 meters in distance, and 30 seconds later is close to 4 meters. So as you can see, does the 6 feet rule work or not? Indeed, it's not. You need to be far apart a little bit. So we equipped with this paper and the simulation model, and we quickly wrote a NSF proposal and get funded. And the aim is to study the bioaerosol behavior to improve ventilation systems in order to prepare the next pandemic, which I predict will probably occur in 10 years. Our hypothesis is that the indoor temperature and humidity can influence pathogen survivalship and behavioral size distribution in indoor environment. So we create this chamber and we nebulize the surrogate pathogens, which we use E. coli. And then we have 3 ways of collection those aerosols, virus-containing aerosols. One is a gelatin filter, an impactor, and also impinger. And then we analyze the pathogens using the colony counting method and also do the qPCR to come up with a ratio, which is a viable DNA divided by the total DNA. And these 2 data sets are basically complementary to each other. We have published 1 paper. We have 2 more manuscripts in preparation. And the take-home message is the pathogen survivalship and bioaerosol size distribution are both temperature and humidity dependent. We also found out that the UV and ozone disinfection can reduce microbe viability, and the outcome is bioaerosol size dependent. That means they do not kill virus E. coli. Okay, it all depends on the droplet size. So the next I'm going to tell you about, so now we know the behavior now. I'm going to switch to the smart mask in terms of how we could protect ourselves. So at the beginning of the pandemic, a person in engineering department came with me. He said, Dr. Huang, we have these 2D materials, and they are MXene and graphene. Can we do something with pandemic? So I think about it, and then I quickly assembled a team with an aerosol expert, with a medical doctor, infectious expert at WashU, and I said, we probably need an electrical engineering person. So we decided to go with the spike protein aerosol testing. And this is our micro fluid device, and you just need to look at this one here. So what we do here is, on top of vaccine, we lay out the MXene, and then APTES is a linker which can immobilize antibodies, and we collect the human saliva, let it flow through, and then once it's binded to the antibody, it will show the change in resistance. So we tested the specificity, and we found that when we use the COVID-19 antibody, it could capture the COVID-19 virus. When we switch to H1N1 antibody, it could catch the H1N1, inactivated H1N1 virus. So with very high specificity. How about sensitivity? For the H1N1, our limit of detection is 125. As long as you have 125 copies per mil, we could detect it. The range is 125 to 250,000 copies of the virus. For the COVID-19, the limit of detection is 1 femtogram per mil, and the range is from 1 femtogram per mil to 10 picogram per mil. So the next step is, I think about, okay, we have this chip. Can we attach this chip to a mask and then let people wear it? And then we could detect the change of the resistance. And this one is basically patent pending. So we collect saliva sample, nasopharyngeal samples, and we found very good correlation with the qPCR outcome. And importantly here, I show you that we ask the patient with the positive diagnosis and let them wear day 1, and then the 3rd day come back, wear it again, and the 5th day. So you could see the declining viral load over time. So we found that indeed we could detect the virus in a non-invasive way within less than 1 second with very high accuracy. And the goal here is, if you are a Jerry Ford aircraft carrier, you won't get stuck in Guam for a month. Remember the story there? Okay, the commander just can dispense the mask, and then let them wear 1 to 3 minutes, and then he could pick up the phone and say, okay, mechanic A, you are positive, you are out, and then turn around to Pentagon and say, I need mechanic A to come to the aircraft shift. Okay, you know, during wartime, 1 hour delay would make the very big difference in the battle outcome. The next one, I think probably my time is almost up. Okay, as an electron signal. So as you can tell, these are all aerosols. Okay, the first 2 stories are tied together. This one is also related to the pandemic, and I'll tell you why. So you guys know about vaping and e-cigarette, right? So there's a significant increase of the use of the e-cigarette over time. They look like this, very fancy. They draw a lot of young people to get addicted to. And this is anatomy of the e-cigarette. There's a rechargeable battery, and you have the e-cigarette here. You light it up, and then this is a coil that basically vaporize this e-liquid, which contain PGVG, nicotine, and flavorings, whatsoever you like. And then after being heated, you have the product there, including heavy metals, mainly coming from the coil, and also you have all kinds of aldehydes. So there are a lot of studies that's been done in terms of cardiovascular diseases. So our goal will be very different because we are not I'm not a respiratory biologist, I'm not a lung person. So what I do here is I found out one thing, that is one faculty member basically screened more than 200 different fabrics at the early onset of the pandemic to come up with the filtration efficiency so that the materials can be picked, can be used for masking purposes. I say, hey, Yue-Wern, can you pick a few of them and come up with a system that will mimic our lung so that we could pass this, the primary or first-hand smoke, and through this filtration set, going through like the exothoracic region, tracheobronchial region, and alveoli region, and then come out of the lungs. So 1, 2, 3, 2, 1, and then it becomes standard secondhand smoke. So 2 months later, he gave me a call and said, Dr. Huang, could you come over? I'm going to show you a figure. So I see this figure, I immediately know, okay, we got the first, the system that can study the secondhand smoke, because all of the studies has been focusing on first-hand smoke, and nobody is able to do the second-hand smoke because they don't have this kind of system. So we wrote it to NSF and get funded, and we found that very quickly, I'm going to write this one up, that is, in general, a person would have 165 puffs a day, and this is a result from 10 puffs. And as you can see, the heavy metals like chromium, manganese, and the nickels, after 10 puffs, the exposure of the patient, of the person, basically is above the maximum permissible limit of the, set by the NIOSH as well as European medical agencies. Okay. So we further do the cell membrane integrity to see whether there's any implication in terms of certain respiratory diseases system. And now we create an animal exposure chamber and try to do the research because people have been focusing on cardiovascular diseases. Now here, I'm coming here to look for collaboration in terms of eye diseases because as far as I know, very, very few, only a few papers publish the eye effect, ocular health. So that's why I'm coming here and try to see whether we could go into this direction, which is really unexplored. Thank you.
Thank you. Your questions? Yes. If you have a question, don't wait for me to call on you. Just come right onto the mic and form a line if you want.
Do you know if there's any plan to kind of go further with this research, maybe look into like aerosol contamination or kind of like occupational hazards with aerosols for like other people's health?
Yes. Yeah, we would like to relate it to the public health. Indeed, there's one research I didn't tell you. I suggest to my colleagues to use a solar house we have on the campus and try to do the second-hand and third-hand smoke experiment. And we have a manuscript in preparation, but I would like to collaborate with medical doctors to address this kind of health issue, including eye diseases. Yeah.
Thank you.
Go ahead.
Hi. Going back to your aerosolizing procedure with E. coli, did you perform that with like viral particles as well? Because I know they're much smaller in size than E. coli, so I didn't know if it could affect the outcomes of your
Yeah, we use E. coli as a surrogate, and we also had a bacteriophage in our NSF grant proposal, and we are also we don't have a biosafety level 3, so we are kind of have limitation. We have this system. If someone has a biosafety level 3, we are more than happy to collaborate using our unique system.
Thank you.
Does Mentimeter have something for us? You're not on. There you go. No?
Can you guys hear me? There
Yes.
we go. This was similar to a question already asked, but can you use your system for pollution exposure research?
Yes, I'm a toxicologist by training, so I did a lot of air pollution and water pollution. So definitely that's an area that's, it all depends on funding, you know that. Yeah.
All right. By observation, in your talking about droplets
Yeah.
and being 6 feet apart
Yeah.
was sort of interesting to me because 5 years ago, this Friday, it was Friday the 13th in 2020, the world seemed to be falling apart. The national emergency was being declared, states were closing schools, national basketball tournaments were being canceled, all on this day. And so I'm glad that, at least for me, now that we've gotten through all that you're still working on that and knowing that 6 feet is still not far enough.
Yeah.
So, all right, thank you very much.
Okay, thank you.